men on the moon. We're trying to organize this thing. It's been like just about as difficult. So thank you very much for the invitation to present here today. The title of my talk is slightly different from just next generation sequencing because I want to put in context how we've used next generation sequencing to direct the development and discovery and development of new therapeutics for endocrine therapy, estrogen receptor positive breast cancers. My group has been involved in the development of endocrine therapies for the past 30 years and been involved in the discovery and development of a lot of drugs, and so do have some inherent conflicts of interest. The most two that are relevant today, and one I'm very proud of, is the elacestrant conflict of interest. That drug was actually developed for breast cancer in my lab. And I'm also going to talk a little bit about lasofoxifene, a drug that was repurposed by a graduate student in my lab that Sermonex now are developing in the Elaine clinical trials. A good starting place for this talk is a general model of how we understand the cancer cell intrinsic estrogen receptor signaling axis, boiled down very simply. There are three basic places where we can intervene. The first, of course, is with aromatase inhibitors. There are 3 approved aromatase inhibitors. There are very unlikely to be any more. The second, though, is the one we're going to focus most on today and is where most therapies are directed against, and that is the estrogen receptor. And then the third, which I'll touch on in a minute, at the end of my talk rather, is the estrogen receptor co-regulator complex, which has become very important given the results of next-gen sequencing that have identified mutations that affect proteins in this complex. But most of the activity actually, as I said, is around here. And what we believed, or at least until relatively recently believed, is that the role of a SERM or a SERD was to first and foremost competitively inhibit the binding of an agonist to the receptor, and secondly then to degrade the receptor, removing the receptor from the equation. If that was entirely true, then when corrected for affinity, all antagonists or SERDs should be functionally equivalent, and clearly they're not. So one of the reasons I was particularly interested in entering this field when I had black hair, I have gray hair now, was because the simple models of estrogen action really didn't describe and inform why so many endocrine therapies, high affinity estrogen receptor antagonists, one after the other failed to make their endpoints. And it was because, I think, we tried to fit them into this simple model, as I said, where agonists activate and antagonists merely function by inhibiting the receptor. I'm not going to go through the many papers and talks by other people at this meeting and others who have described how we've evolved from that simple model. But this is the model that my group mainly has put together over the last 15 years. And there are 3 points that I want to bring across in describing this model that will help us understand the differences between existing SERMs and SERDs. The first is the receptor does not switch between an on and an off form. What we now know is that antagonists can drive the receptor into one conformation clearly, pure antagonists, and agonists, in theory, drive the receptor into a conformation at the other end of the spectrum. But in work that was done initially by Andy Shao and Jeff Green and Rob Hubbard, it was shown that drugs like tamoxifen don't really freeze the receptor in an inactive conformation, but drive the receptor in intermediate conformations. And from that work, and then work by John Norris and my group over the years, we discovered that subtle changes in the structure of the chemical of the estrogen receptor modulator can have profound effects on the shape of the receptor. I'll always remember when we submitted this paper for review to Science in 1999, the reviewers came back and said, the McDonnell laboratory has developed a very sophisticated technology to detect microallosteric changes in the estrogen receptor of unlikely importance. And so for those of you who have heard the reviewer number 3 stories, most of them are true. The second lesson, though, that we learned is that shape matters, and it matters because it allows the receptor to engage different co-regulator proteins. And the third rule, then, is that cells differ by the expression and activity, relative expression and relative activity of the attendant co-regulators in cells. So taking that information together, we came up with a hypothesis several years ago, and that was when, in about the late 2000s, when tamoxifen resistance was what drove research as opposed to aromatase inhibitor research. What we ended up demonstrating is that tamoxifen functioned as an antagonist by driving the receptor into an inhibited form. Cells just couldn't recognize it. But we hypothesized, and I think it's now been shown to be true, that receptors adapt and are able to then find co-regulators that can interact with this aberrant structure of the receptor, and the tumors progress. And so a graduate student in my lab, Caroline Connor and John Norris, who at the time was a postdoc, came up with this hypothesis that if we could select for compounds that would drive the receptor into yet a distinct or a different shape, you would disengage this cofactor and you start the process all over again. Well, to make a long story short, Caroline and John ended up discovering a molecule called DPC-974. It was named Taxol. And this drug was the very first oral SERD. I'll show you why that is in a second. This drug was found in a defunct drug discovery program for osteoporosis, of all things, in GSK. This, I will show you in a second, it drove the receptor into a different conformation, and in models of tamoxifen resistance, here are MCF7 ER-positive breast tumors growing in the presence of tamoxifen, that's classical resistance, it inhibited the growth of the resistant tumors. Jeff Green and his team then, several years ago, actually proved out of molecular detail the extent, or really probed the extent to which this molecule really did affect the structure of the receptors, really helping us to prove the hypothesis that conformation matters, by showing that in green here is the crystallography of the hormone binding domain of the estrogen receptor, and in yellow the tamoxifen structure. And you can see that there's a very dramatic difference here at one of the pockets in the receptor that is important for co-regulator binding. So it turns out that conformation mattered, and that by selecting for molecules that drove the receptor into different conformation, we could actually get molecules that would inhibit the growth of tamoxifen-treated tumors. But we got really, really lucky. Okay, I'm Irish. Irish people have a lot of luck. And the reason we got lucky was because Ashini Wajaratne, who was a graduate student, noticed that this molecule, DPC-974, was in fact a degrader. It degraded the estrogen receptor. These are ER-positive breast tumors. And you can see estrogen downregulates on receptor, tamoxifen stabilizes the receptor, fulvestrant degrades the receptor. And this molecule, DPC-974, degraded the receptor, and this is the quantitation. This was allowed us then to kind of update our model slightly. We knew that first and foremost, this molecule, and like all of the other SERDs, all of them are first and foremost competitive antagonists, blocking the binding of an agonist. But it turns out that the conformational state that some of these molecules drive the receptor into it, targeted for proteasomal degradation. So what happened to DPC-974? It was ahead of its time. It went into clinical trials. DuPont and BMS brought it into clinical trials. And Patrick Neuven over in Leuven showed that it actually worked in heavily treated patients. And then something happened. Some young whippersnapper MBA decided that there was no room for endocrine therapy anymore after aromatase inhibitors, and the entire field stopped. And it wasn't until several years later that people came along and, building on these concepts, built the next generation of endocrine therapies. Never trust MBAs. So why is it a good thing? And this may be what reinvigorated the field. Well, Benita Ketsalone-Bogan, Ken Korak, and Bert O'Malley, all at different times, and many others, discovered that the estrogen receptor does not always need to be activated by a small molecule ligand. And what they found is that activation of different signaling pathways in the cell could lead to hyperphosphorylation of the receptor at very specific sites associated with receptor activation, or in fact, even as importantly and possibly more importantly, phosphorylation of the receptor coactivators. And so what you can see is that you can imagine is that taking the receptor away basically negates all of these ligand-independent pathways. And I think that this is really the one slide, if you want, that tells you why SERDs may actually have different activities over SERMs in some circumstances. It's a very crowded field. And this is I took my prerogative of placing compounds along this axis between SERMs and SERDs based upon the profiling that we have done over the last few years. The DPC-974, which is the molecule I just described, is chemically very similar to all of these molecules here. There's a large group of them. Somebody mentioned that there are 10 drugs in clinical development. There are actually 13 right now in clinical development in this class, so it's a really busy field. At one end of this field spectrum is fulvestrant, and there's an older compound from Roussel-Leclerc. In my opinion, nothing comes close to the degree of degradation that those molecules can achieve, but they both have PK issues. You'll notice I put elacestrant here around 1901. As I told you, that drug came from research in my lab. We never called it a SERD. We called it a SERM-SERD hybrid because it really doesn't degrade the receptor very well, only about 50%. And what that says is that although we try to bring things down to make them very simple, these molecules are not quite simple. That drug is working probably because it's a good competitive antagonist, it's a good degrader, but there are other activities we believe in the immune system that actually make this drug particularly useful. And then the drug I'm going to mention out here in a few minutes, lasofoxifene, would be a classical SERM, a molecule that has protective activities in bone, functioning as an estrogen, but also has an interesting clinical activity. So that leads me to the first story that we need, or a couple of series of slides we need to talk about what the products of next-gen sequencing are. And of course, you want to be on the planet Orb not to realize that ESR1 mutants have become a cottage industry within endocrine therapy, identifying them, profiling how compounds interact with them, and trying to understand the biology underlying the different mutants. I don't want to go over this too much detail, and just to say that between 30 and 40% of endocrine therapy-resistant metastatic lesions contain at least one ESR1 mutant. The question is, what is the importance of these? And in 2 slides, I want to just introduce 2 concepts to you. The quick and easy answer is that these mutants contribute to the biology of the tumor because they activate the transcription absent an estrogen, and that is absolutely true. Here is just a transcriptional assay showing the activity of a wild-type receptor in response to increasing concentrations of estradiol. And here is 2 of the most commonly found mutants, the 537S and the Z538G, and you can see they're constitutively active. So clearly that's the case. You can see why fulvestrant may not be as effective against these mutants because these mutations shift the affinity and actually decrease the potency, shift the potency by decreasing the affinity by about 1 log. And so you say, well, let's go ahead, this is a simple pharmacological problem, let's just increase the dose by 10-fold. We can't do that. We're right on the edge with fulvestrant. This is the Radius compound. As we know, it works in ESR1 mutants. Yes, its potency is also shifted, but we can deliver enough of that drug. But this was lasofoxifene. So this is a drug that I was involved in development back in 1991. I definitely had black hair then, when I was working for a company called Ligand Pharmaceuticals, and we were developing SERMs for bone. And it was in our collection when a graduate student of mine about 4 years ago decided to look at all the ESR1 mutants. And what she found is that the mutations had no effect on the binding of lasofoxifene. And that was enough to get David Portman and his colleagues to actually start the ELAINE trials. And I'm looking forward to the output from those trials. There's one other thing I want to show you here. This is one of the original Genentech compounds, GDC810. And it's going to be relevant to a slide I'm going to show you. That compound, you can see it clearly inhibits the wild-type receptor. It's a SERD, by the way, I beg your pardon. It degrades about 97% of the receptor, but not all of it. And you can see that compound inhibits the wild-type receptor. But the 530S mutant, not only does it not work, it's actually activated by that compound. So that leads me to this one slide that looks complicated, but it's not. And it's a slide where I want to drive home the point that don't be thinking about compounds that are hitting SERDs as just needing to bind and to inhibit estradiol binding to those mutants. Because what we're finding now is that depending upon the ligand that binds to each of these mutants, we are creating neomorphs. So the combination of the mutation that is evolved in the aromatase-resistant patients, plus the added conformational change that's been added by the SERM or the SERD, is actually creating sometimes beasts. And let me show you this. This is a fingerprint of the structure of the compound. This is using peptides that decorate the surface of the receptor and pepper it so we can look at conformations. And we do that, what you can see is that clearly the binding pattern of these peptides, in the presence of beta-doxyphene on the wild type, is completely different to that if you look in the presence of the 537S or in the presence of 538. So what does this mean? It means that these compounds, these SERMs and SERDs, are likely to manifest different activities, not broadly across, not only broadly across the mutations, but also with respect to each individual mutation. That neomorphism scares me a little bit because I don't quite understand it yet. The second thing that next-generation sequencing has done, and most of this has actually come from Matt Ellis's lab, is identified fusions. And these fusions they contain the estrogen receptor, the DNA binding domain of the estrogen receptor, but the ligand binding domain is gone, and you have all of these other proteins that are fitting in there. And so what this basically suggested to Matt and others is that the DNA binding domain will still allow the receptor to bind to its targets, to its normal estrogen response elements, but its cargo may actually allow the receptor to have different types of transcriptional activities. I'm going to show you one example of this. And this is a T47D xenograph that Matt and colleagues engineered to express 3 of these different fusions. Okay, the YAP, this one here, and NOP2, I can't see it from here. But you can see that whereas normal T47D cells require estradiol to drive their proliferation, in the absence of estradiol, you get varying degrees of growth of these tumors that express these particular fusions. So they don't bind to the ligand binding domain, so this is problematic. But Matt did something very smart. What they did was they moved down below the receptor and said, well, what's the first step, growth-promoting step, below the estrogen receptor? Well, as we all know, it's the CDK4/6 or B axis. And so he made the assumption that these mutations then should succumb to CDK4/6 inhibitors. And so here's a case of a tumor that is endocrine therapy resistant, but clearly, and because it contains this YAP mutation, but you can see that clearly what happens is that palbociclib will inhibit this tumor. So what's happening is we've lost the control of this CDK4/6 axis by the estrogen receptor, but by jumping down, inhibiting the CDK4/6 with palbociclib, we can actually tackle this. Matt's taken on a second approach, which I like, and that is to make the assumption that these mutations or these fusions are conferring vulnerabilities upon cells that can be targeted. And they've identified pathways in some of these mutants that can then be targeted with kinase inhibitors, for instance. A third thing, and I don't expect anybody to really be able to read this slide, Corinne Haynes, who's a postdoc in my lab, dug through the literature and went and looked for mutations in co-regulators. And it turns out that between 1% and 2% of all cancers have mutations in co-regulators, and they're likely to be important also in response to endocrine therapy. In fact, Kent Osborne's group, in collaboration with Bert O'Malley's group years ago, showed that SRC3 amplification, the co-regulator, was associated with increased resistance to endocrine therapy. And so these are also very problematic because they're not classic druggable targets. I don't like to use the word undruggable anymore because I think everything is druggable. You just need to work harder. But classically, these are not druggable targets. But I want to just show you what I think is very exciting. So several years ago, about 4 or 5 years ago, and it was more than that, 6 or 7 years ago, Bert O'Malley proposed the idea that you could be able to come up with small molecule inhibitors that would inhibit the various co-regulators by binding into nooks and crannies in these proteins and inhibiting their activity. I swear to God, people thought the guy was nuts because these were considered to be undruggable targets. But he actually identified a whole series of molecules and has published a lot of them. And I think that we're going to see a future for these, not only in estrogen receptor positive breast cancers, but also in other cancers. To make a long story short, Bert ended up finding these things called SMIs, small molecule inhibitors of SRCs. And what he went into an SRC3 overexpressing xenograft. And what he found was, no big surprise, that the co-regulator binding inhibitor alone didn't do very much because the estrogen receptor is still there, can interact with other proteins. But when he combined it with a SERD, AZD9496, which has since been continued, but it's still a good tool compound, they were able to inhibit the growth of this. So what this suggests then is that, where we were talking this morning about de novo resistance to endocrine therapies, I think that what we're going to find is that some of those de novo resistance mutations, so those mutations that lead to that de novo resistance, are likely to be mutations in co-regulators. And whereas the co-regulator inhibitors may not have single agent efficacy, they may clearly synergize with SERNs and SERDs. And then the final thing I want to do in 2 slides is just introduce a concept that I'm not sure too many people know about. And that is, we talk about aromatase inhibitors inhibiting endogenous estrogens, and that's the way they work, and clearly that's the way they do work. But what not very many people here, I bet you realize, is that we all have in us a vestigial estrogen called 27-hydroxycholesterol. It's a primitive estrogen that is used in lower eukaryotes, and we still make it. But although we don't know what the physiology of this hormone is. It's made in a single-step transformation from cholesterol to 27-hydroxycholesterol. And Eric Nelson, who was a postdoc in my lab, he's now an associate professor at University of Illinois in Urbana, showed that he really did the definitive test to prove that this this endogenous ancestral estrogen is important by taking endocrine therapy-resistant tumors and showing that they won't grow without hormone, they grow in the presence of estradiol, and they grow in the presence of tamoxifen, as I told you. So they're able to, the growth is stimulated by those. That's classical endocrine therapy resistance. But look what happens. It's stimulated by 27-hydroxycholesterol. So we did a clinical study to ask with Singo Borquist at University of Aarhus in Denmark and asked a very simple question. First of all, is there enough of this molecule in humans to activate to activate the estrogen receptor and support the growth of the tumors as we had shown in animal models? And the answer is absolutely. Shown in light blue here are the levels of 27-HC that are required that we have found in animals that are required to support the growth of these tumors. And you can see here two things. One is the levels of 27-hydroxycholesterol in most of the patients we looked at were way above that. The second thing which we did was established a pretty good relationship between total cholesterol levels and 27-hydroxycholesterol levels. And that suggested to us, let's go and do another clinical study. And what we did was we took breast cancer patients in a window of opportunity study, and we just treated them for 28 days with atorvastatin and asked, obviously we're going to be able to lower LDL cholesterol, but could we lower 27-hydroxycholesterol? And the answer is we did. I don't pretend to say that this is the only reason the statins work in cancer, but it's a pretty good one. It gets rid of an endogenous estrogen. And so I'll finish up then by just coming back to the points that I tried to make at the very beginning. I think I glossed over this purposely by saying that, we know that aromatase inhibitors block the production of estrogens. That's steroidal estrogens. They don't block the production of 27-hydroxycholesterol. That's made by a different enzyme, CYP27A1, and we have now developed inhibitors against that. The second thing is, is that the estrogen receptor is a target, will remain a target for a long time, but please throw out of your mind the idea that it's an on or off, that all you need to do is turn off the receptor. We need to get rid of the receptor if we can, and we also need to remember that these ESR1 mutations in the context of current drugs we have may actually cause unexpected neomorphisms. And then finally, we need to focus on the co-regulators, and hopefully that we'll be able to see the advent of co-regulator inhibitors coming into the clinic. I'm going to skip that. So then let's just finish up with a couple of points. I still think for now, absolute inhibition or degradation of the estrogen receptor within cancer cells, and I stress within cancer cells, should remain a target. I think that we need to be able to inhibit 27-hydroxycholesterol. I think all high-affinity antagonists will do that. And I think that some of the ESR fusions may yield to conditionally induced vulnerabilities or some of the novel PROTACs that are being developed, not the ones that are targeting the ligand binding domain, but other regions in the receptor. And I skipped over this because I was hoping to get an invitation next year to present it. But we just published a paper in JCI the other day showing that as important the activities are in targeting the cancer cell intrinsic actions, what people have forgotten, and we've now demonstrated, is the cancer cell extrinsic actions of these are very important as well. We've shown that SERMs and SERD have completely different with respect to their impact on myeloid cell biology and T cell biology. Secondly, we forgot the brain. And what we now have data to show that SERD activity in the brain is not what you want. In fact, we've been able to show you can make tumors completely grow independent, completely in the absence of estrogen by knocking out the estrogen receptor in certain regions in the brain. And that's a complete 180-degree turnaround for me personally because I wanted to get drugs into the brain the whole time. So why does elacestrant work in the brain? It's a partial agonist in the brain. And finally, when we're we're talking about these drugs, we need to talk about the adjuvant setting and going forward. And there are some other things that we need to talk about other than the sequelae associated with bradycardia, ocular disturbances, and neutropenia. We also need to understand that these SERMs and SERDs are going to differ in the way they protect bone. Osteoporosis is equally morbid a disease in the age population, and we don't want to create a worse disease than we're trying to cure. So thank you very much, and I hope those comments were useful to you.
Thank you, Donald. That was extremely interesting. So the next speaker